Thank you so much, Lionel. Muchísimas gracias para tenerme acá. Thank you so much for the invitation. I'm really excited to talk to you today about fluorescence lifetime imaging. Es un placer de estar aquí con todos los estudiantes. Hola a todos. Okay, entonces voy a empezar. I'm going to start now. Can you see my slides just to be sure?
Yes. Yeah.
Okay, great. Thank you. Okay, so today I'm going to talk about an application to metabolic imaging in tissues. And one of the applications that I'm really excited to talk to you about is cancer research. So as you all saw earlier from the workshop, that the Phasor analysis is a very powerful way to graphically represent fluorescence lifetimes. In fact, you could see the fingerprints of any fluorescent molecule on the Phasor diagram here. And these are just a few. Some of these are really important in tissues such as retinol, FAD, arachidonic acid, retinoic acid, free NADH and bound NADH. And one of the things that makes this particular graphical representation exciting is the fact that you could actually excite the autofluorescence, the intrinsic autofluorescence in living tissue. And specifically, what's important about this is the nicotinamide adenine dinucleotide fluorescence or autofluorescence. In fact, this cofactor, this coenzyme actually plays a critical role in the energy metabolism. So as you could see the structure, when it's bound to a protein, the complex is open, so the fluorescence lifetime is very long. Depending on what it's bound to, it can have a lifetime up to about 9 nanoseconds. In the case that we use it, we see fluorescence signal at 3.4 nanoseconds. When the protein dissociates or comes off of the cofactor, these two, the two moieties at the end of the structure actually come together and quench the fluorescence signal, so you get much less intensity here. The quantum yield goes down, and then the fluorescence lifetime is very short. You get in the order of 0.4 nanoseconds. So the information about the protein bound and free can actually serve as a metabolic let's say, marker for what type of metabolism the cells are undergoing. And one of the stories I'm going to show you just, this is one of our outreach programs that Lionel was talking about. I usually recruit all of our postdocs and students to do community outreach for community college students, specifically URM, what we call here underrepresented minority students. We did this kind of experiment where we took macrophages. And macrophages can differentiate into two different cell types, the M1 macrophage cell type and the M2 macrophage cell type. And if you are an avid, let's say, supporter of the Star Trek or Star Wars, you will notice here that this is the force, it's a balance as to when macrophages become differentiated. So M1, for example, are cells that are kind of toxic. They stimulate cytotoxicity and tissue injury, so they're on their dark side. And if the macrophages differentiate to M2, they're the Jedi order, and they have immune suppression and tissue repair. So we took these macrophages and looked at the autofluorescence signal. And the first thing to do when you're doing these kinds of experiments is to characterize the free and pure, let's say, component of the possible fluorescence lifetime you have in your pixels. So you can take NADH, you can buy this from Sigma, dissolve in PBS, and you'll notice that the lifetime here is very short. And with the phasors, we can just graphically calculate, let's say, represent where the distribution of these fluorescence lifetime signals are. So we have here free NADH, and they're all red pixels. The bound NADH, which has a much longer lifetime, and anything that is in between is just a fraction of one to the other. And what you'll notice here is that macrophages have a heterogeneity in distribution of free and bound NADH. Mostly in the nucleus is more free NADH, and that's true of all cell types, honestly. In the cytoplasm and mitochondria, that's where NADH becomes more bound. And so for this study, we looked at M0 macrophages and compared them to M1 and M2 macrophages. And we can actually differentiate these two distinct macrophages using different biochemical components. And with the color pixels here, which I didn't talk about, so more purple is a higher fraction of bound NADH, more red is a higher fraction of free NADH. And we can characterize here, just looking at the colors, you'll notice that M1 macrophages, which were the dark order proteins, let's say characterizations, have more yellow pixels. And yellow here is higher free NADH, purple is being more bound. So distinctively, you can already see this. And if you quantify just the percent color, and remember, these are community college students, so we did this in a week, you'll see that M1 distinctly has a more free NADH. And free, I'm going to talk to you about the glycolytic metabolism. I think Suman may have mentioned during his talk as well, of these different types of distinct free to bound fractions. But you can see clearly that M2 have a higher bound NADH, and that's indicative of oxidative phosphorylation. So one of the things that made the Phasor plots so powerful was the fact that if you have, let's say, a characterization of many possible lifetimes, and now we can do a lot of cluster analysis to distinguish which component is giving you the fraction of each one of these types of molecules. But at the time, we were getting these plots that look pretty much like a blob on the Phasor. And that was really difficult to discern where the signals were coming from. So just to give you kind of a historical perspective on how we were able to, let's say, dissect this Phasor plot, and we were able to, let's say, take an image, and let's say that you have 3 different cells. And if you take the image here, and this is your fluorescence lifetime, these can be independent cells, or they can be components inside the cells, like organelles. And if you take all of this data and plot it on the Phasor plot, you'll see this blob, right? So it's very hard to distinguish this, let's say, object from this object from this object based on this distribution here. But of course, if you are able to characterize, let's say, the average fluorescence lifetime, or the average, let's say, Phasor lifetime for each one of these individual components, we can now have a way to distinguish the degree of separation from each one of these components. They could be different cells, or they can be in internal organelles, for example. So in this case, we did what is called the Phasor, cell Phasor distribution. And I'm going to show you some examples of this, and also how did we know that this metabolic trajectory on the Phasor plot was indicative of glycolysis and oxidative phosphorylation. I think Suman gave a very beautiful talk about one of the early works of Britton Chance in looking at NADH autofluorescence and looking at the metabolic states within the fraction of those free and bound NADH. So let's take a look really close at what's happening in cellular metabolism. So our cells can actually break down glucose in 2 different ways, either just normal cell respiration, which is called oxidative phosphorylation, and that's using the mitochondria, or they can break down glucose in the absence of oxygen. If your muscles, let's say you go to the gym, you work out a lot, your muscle cells can actually undergo glycolysis, producing a lot of lactate acid. So what you see here is that in this glycolysis pathway, there's a lot of NADH that is being produced. This is an NADH producing pathway. In the oxidative phosphorylation pathway, we also have NADH producing, but also it gets very much bound to specific enzymes during this, let's say, during the Krebs cycle and electron transport chain, specifically here in the electron transport chain. So this is called the NADH consuming mechanism for cellular respiration. So again, just to summarize, we have cells that can break down glucose in the presence of oxygen. This is normal oxidative phosphorylation. It makes 36 moles of ATP. And in this case, we have a low free NADH ratio to more bound NADH ratio. Of course, cells can also break down glucose in the absence of oxygen, and this is called anaerobic glycolysis, producing ATP. and also producing a bunch of lactate, which is not good for tissues. However, what's characteristic of tumor cells and proliferative tissues, such as stem cells, is the fact that these cell types don't need to have oxygen. They can revert, they can switch between glycolysis and oxidative phosphorylation. And here they produce 4 moles of ATP. This is known as the Warburg effect, essentially. So tumors have a preferential metabolic pathway due to the fact that they need to divide, they need to proliferate quite quickly. Even though they produce less ATP, this pathway is much faster. So in this case, they could actually produce ATP at a faster rate than oxidative phosphorylation. So one of the ways to characterize this shift on the Phasor plot, and many other people have worked on this, Melissa Scala's group has worked on looking at free and bound NADH. But what's nice about the Phasor analysis is that you can see this shift very easily. So here we have the, let's say, cells. We can add potassium cyanide, which is an electron chain inhibitor. And so we're actually targeting the metabolic pathway through the mitochondria. So this is inducing the cells to undergo production of ATP, but in the glycolytic path. So you'll see that these pixels are more free, more pink. They're more free NADH compared to normal cellular respiration where there's more bound NADH. You can also revert the metabolic pathway. You can make them, can you see me still? Okay. So you can make them revert from a more free NADH glycolysis to a more bound NADH using hydrogen peroxide. So there are actually a lot more inhibitors that you can buy commercially. One of these inhibitors are dichloroacetate. This shuts down the glycolytic path. So dichloroacetate is actually used in chemotherapies because the idea is to take cancer cells that are highly glycolytic, highly proliferative. You want to shut down that mechanism by targeting their metabolism. And by shutting this pathway down, the glycolytic pathway, the idea is to revert them to a more oxphos state. And they believe, cancer biologists believe, that if you revert their metabolism to an oxidative respiratory stage, you can actually tune down their proliferative aggression. So they become more quiet, let's say, undergo more quiet state. You can also target the mitochondrial pathway, oxidative phosphorylation, using Rotenone, Antimycin A, and these are inhibitors of the oxidative phosphorylation. Rotenone binds to complex 1, Antimycin binds to complex 3. And if you're going to do these experiments, I always recommend using both Rotenone and Antimycin A to make sure that you're shutting down this pathway of respiration. So in this case, we have, you can see very clearly, if you take cells and you incubate them in 4 millimole, that's 4.5 millimoles of glucose, very low glucose versus very high glucose concentrations, you'll see that you can switch their metabolism quite easily. So the cells become more glycolytic. They go from oxphos, normal respiration, to a more glycolytic state. You have more glucose in there, and they become highly glycolytic. And under dichloroacetate, remember, this is a drug that targets cancer cells. These cancer cells are highly glycolytic. You see more pink pixels here that are indicative of free NADH, and you can revert them to a more cellular respiration, slowing down their proliferation, their cell division. Okay, so here is now a final treatment with Rotenone, Antimycin A together at about 10 micromolar concentration, and these cells are shutting down their normal respiration. They're shutting down the oxidative phosphorylation path and reverting to a more glycolytic pathway. So I'm going to show you some in vivo experiments that were done with collaborators here at UC Irvine. And this work was done together with Chiara Stringati, who is now in in France. She was a postdoc in Enrico's lab, and we worked together in this project, and we took these gastrolumen sections. So this is part of the small intestines. And what we did is we had to cut them so we're able to image into this part of the tissue. And if you look at the objective here, you could see that we can either image from the bottom into the tissue or from the other side. And what's unique about this tissue is that there is a stem cell niche right here that is really important for the differentiation of the cells that form this villi structure. And so if we look more closely into the structure, you'll see the stem cells are down at the bottom here. And as they differentiate to the top, their metabolic switch can happen. So first, we're identifying the stem cells. Where are the stem cells? We used a transgenic animal to excite GFP. This is at 880 excitation. You could see the collagen structure in the tissue. And first, if you look just at the blob here of the Phasor plot at 740, you see a distribution of free and bound NADH, and then you get this distribution of autofluorescence. So first, we had to identify where the stem cells were. You could see the GFP identifying here, and then the free and bound NADH here. You could see that these cells are very, very glycolytic. They're pink, they're more free NADH, and the cells surrounding here are more bound. And those cells, if you colocalize these, are stem cells surrounded by something called the Paneth cells. And you can take different Z sections of the intestinal wall, and if you do a cell Phasor average, you'll see that the Paneth cells are more on the right side of the Phasor plot compared to the stem cell, which are more to the left. And each one of these spots is a single cell. And you can do Z stacks, because if you want to look at the differentiation along this axis, it's important to look at the cross section of the lifetimes. And in the Z Phasor plot here, what you could see is the average lifetime at the bottom of the crypt. Here, they're more glycolytic. And as we move up to the crypt, you get more bound NADH. So finally, this work showed that we can take and differentiate the cells in the tissue from Paneth cells, stem cells, proliferating progenitor cells, and terminally differentiated stem cells up at the top where they have this microvilli structure. And this correlates really well with Wnt signaling. So the last part, I'm just going to take 1 minute to show you this really important work that we did on Wnt signaling. This is the work by Marion Waterman, and she's interested in angiogenesis of cancer cells. So if Wnt signaling goes wrong, then the cancer can start in the colon. So they think that there's a protein called PDK1 that is regulating oxidative phosphorylation. It's inhibiting OXPHOS, going to glycolysis and causing angiogenesis. So there's a correlation between glycolysis and blood vessel growth because of this protein PDK1. So they actually have a transgenic model where they express the dominant negative left TC form that lacks the beta-catenin binding domain. And so this is a dominant negative that will allow OXPHOS to occur, meaning that if you take away PDK here, then oxidative phosphorylation will happen. You will not get angiogenesis. You will not undergo glycolysis. So pyruvate dehydrogenase, which is the PDK1, is playing a primary role in inhibiting the pyruvate flux to mitochondrial respiration. So they have these transgenic mice. I don't have time to go into that, but what's really important here is the data. So if they don't express PDK1, if you don't express the dominant negative, meaning the inhibitor of PDK1, you could see the cells are highly glycolytic. There's more free NADH. Now you add DOX to express the double negative left, which inhibits PDK1 expression. And now you see that the cells go from glycolysis to OXPHOS. So you've switched the metabolism in these cancer cells to more oxidative phosphorylation. And you see the data here, each one of these is one single cell. And in the presence of DOX, you inhibit glycolysis and they're more bound, more OXPHOS state. You can rescue it back by adding PDK1 into the tissue, and you'll see that you get back glycolysis. And you can see together how this affects angiogenesis, and I don't have time, but I'm going to show you the final data here. So you could see here, this is the normal cancer cells. This is, so in this case, this is the control. The cells, FLIM signatures here, and you could see high angiogenesis in the perfuse vessels. Now, if you shut down PDK1, you could see that with the double negative left, the dominant left, you see no angiogenesis. So the blood vessels were not allowed to grow into the tissue because you've knocked down PDK1 that is inhibiting OXPHOS. So you can now add PDK1 back and you'll see highly glycolytic cells. So this is OXPHOS, which is good. You don't want these blood vessels to supply blood to the tumor, nutrients to the tumor. But now if you add PDK1 back, you get tumor, you get glycolysis, and you get angiogenesis. So basically at the end, with this Phasor approach, you can really start to target cancer cells by inhibiting their metabolic state. In this case, we found that PDK1 was one of those enzymes that was really crucial in switching the metabolism, that if you take it out, you can actually inhibit glycolysis, inhibiting blood vessel growth. So that's the whole idea, is that you want to make sure that you inhibit this.